therapeutics using qFRET technology. So Nikki, take it away.
Okay, hi, I'm Nikki Wong from Dr. Liao's lab, and today I'll be presenting on screening E3 ligases in human genome to identify novel target for anti-influenza virus therapeutics using qFRET technology. So first, to give a little background, the influenza virus has been responsible for many various widespread pandemics and epidemics, such as the Spanish flu of 1918 and the pandemic of H1N1 in 2009, with influenza A virus, IAV, being known to trigger most of the outbreaks. The National Foundation of Infectious Diseases has estimated that the flu cost businesses and employers $11.2 billion in direct and indirect costs, and the World Health Organization has estimated that there has been approximately 1 billion infections, 3 to 5 million severe cases, and 300 to 500,000 deaths annually. However, despite the strides for the development of virus technology, the efficacy remains suboptimal, especially against the IAV variants, as the virus mutates quickly. The influenza virus continues to evolve and develops drug resistance to the already existing therapeutics. It was discovered that the viral proteins hijacks the post-translational modification pathway SUMOylation to replicate itself and block the immune response of the host protein. Similar to how the viral protein can utilize the SUMOylation pathway, the host protein can also offset the effects by modulating its immune response through SUMOylation. So the objective of the project is to discover the E3 ligase in human genome, which is responsible for the SUMOylation of the viral protein, therefore identifying new drug target for anti-flu therapeutics without drug resistance. So SUMOylation is a dynamical process directed by sequential enzyme cascade consisting of the E1 activating enzyme, E2 conjugating enzyme, and E3 ligase, in which SUMO molecules are covalently conjugated to the lysine residue of the target substrate. SUMOylation can only occur on the lysine residues. As seen on the figure on the screen, all the sites labeled red are lysines in the M1 sequence that can be SUMOylated, with there only being 9 binding sites in the M1 sequence. The SUMO molecule will bind to the lysine, removing one hydrogen to form an isopeptide bond. And from previous research, it was found that the M1 can be SUMOylated, but it is still unknown which E3 ligase can bind to it. So using the quantitative Förster resonance energy transfer developed in our lab, we can characterize the interaction affinity between E3 ligase with the IAV virus. Förster resonance energy transfer, also known as FRET, is a powerful phenomenon in which we can analyze the interactions between two proteins that have been tagged with fluorophores. The two fluorophores, when placed in proximity of 1 to 10 nanometers, will induce an energy transfer from the FRET donor to the FRET acceptor through dipole-dipole interactions as depicted on the figure on the screen. Quantitative Förster resonance energy transfer is a technology that can then assess the protein interaction affinity, KD, which can help us to better understand the binding affinity to mitigate the interactions between the two. So the first step performed is molecular cloning, where the PET28B plasmid encoding the fluorescent CY-PET and Y-PET were cloned into E. coli using varying strains of electroconfidence cells using electroporation. Then the E3 ligases were paired with CY-PET, and the IAV viral M1 protein was paired with the Y-PET. After an extensive screening process, the highest strains were identified for protein expression. Then the proteins underwent metal affinity chromatography as their proteins were histidine tagged and exhibited a high affinity for nickel. So using nickel beads and wash buffers, our desired protein was acquired, and high concentrations of imidazole is used to release the his-tag protein by competitive inhibition. Then the eluted proteins were left to dialysize overnight to remove small unwanted compounds from the macromolecules, and a concentration was determined by measuring the fluorescence intensities at their respective excitations. Then a preliminary EM-FRET assay was performed using a 1-to-1 micromolar ratio of CY-PET and Y-PET fused with the E3 ligase and IAV M1 respectively. The alpha and beta values necessary to calculate the EM-FRET were prepared accordingly, with the alpha values prepared with CY-PET alone to account for the emission peak at 475, and beta values were prepared with Y-PET alone to account for the emission peak at 530 nanometers. A qualitative spectrum graph was also performed to visualize the energy transfer between the two proteins at 0.1, 0.5, and 1.0 micromolars, and a KD was determined by keeping the donor protein at a constant of 0.1 micromolar and titrating the acceptor protein concentration from 0 to 25 micromolars. Then, using a software called PRISM, we inputted the calculated qFRET values and placed a nonlinear regression fit line to obtain the KD value. So these are my results. The FRET spectral analyses were generated prior to KD and qFRET determination at concentrations of 0.1, 0.5, and 1.0 micromolar, aimed at examining the specificities of the binding between the E3 ligase with the IAV M1 protein, as well as to analyze whether the interactions are concentration dependent. So the graphical data offers a qualitative visual representation of a select set of E3 ligases, and an increase of the substrate loading shows a clear energy transfer between a pair at the 530 nanometer emission peak. So a quantitative FRET signal determination will help to provide more information into the binding nature between the E3 ligase and the IAV M1 protein. All the E3 ligases and M1 proteins were evaluated at a 1-to-1 micromolar ratio and excited at wavelengths of 414 nanometers and 475 nanometers for CYPET and YPET respectively. So the values were inputted into a bar graph, as we can see here, to better visualize which interactions exhibited a high qFRET value to compare with the spectral graph. So consistent through the spectral graph and the qFRET determined data, HTRIM28 and PIAS1 has shown a clear energy transfer at 1.0 micromolar and a high qFRET value of 653 RFU and 565 RFU respectively. So hence we chose these two proteins for KD determination. The KD was determined by quantifying the absolute FRET signal between the interactions of the two proteins. And to our surprise, the HTRIM28 protein, although exhibited a clear energy transfer and had a high qFRET value of 653 RFU at 1 micromolar, had a high KD value of 24.2 micromolar, meaning the IAV M1 protein does not have a high affinity for it, whereas the PIAS1 protein stayed consistent with the preliminary data and with a high qFRET value of 565 RFU, had a KD value of 2.9 micromolar, meaning that the IAV M1 protein exhibited a high affinity for PIAS1. So the results presented showed the three different methods of FRET, spectral, qFRET, and KD, all exhibited varying degrees of affinity, and the two methods of spectrum and qFRET suggested an interaction, but the true interaction is determined by the evaluation of KD, and the reason supporting the variance is due to the presence of nonspecific binding between proteins, which can be observed when high concentration can force two proteins to interact spatially rather than by its true interaction affinity. It is especially true for large-sized proteins like HTRIM28 with a size of 145 kilodaltons and at a concentration of 1 micromolar, since the measurements were conducted in a 384 well plate. qFRET can solve this problem as the FRET signals for the true target protein interactions were calculated by subtracting the interference of nonspecific interactions and non-target protein interactions. Then, by increasing the amount of YPET IAV M1 protein and keeping the HTRIM28 concentration constant, the binding between the two target proteins will reach a saturation point where no more binding can occur. So hence why the KD value can provide the most accurate results. So the future directions for this project will be to assess more of the KD for the E3 ligases that had high qFRET value at the 1-to-1 micromolar ratio, as well as to check the interactions between the various E3 ligases with E2 conjugating enzyme UBC9 to determine whether it is a real SUMOylation E3, to develop chemical inhibitors for the development of antiviral therapeutics. And SUMOylation assays will also be conducted to determine whether the SUMO E3 ligase helps with the conjugation of SUMO peptides on the substrate. So in conclusion, qFRET technology is powerful to identify SUMOylation E3 ligase for IAV M1 protein, as evident from the results, and genome-wide search for SUMOylation E3 ligases for IAV M1 protein is both important for research as well as for the development of novel antiviral therapeutics development. And I would also like to extend my gratitude to my faculty mentor and advisor, Dr. Liao, for his guidance and support throughout the entirety of the project, as well as to all my lab members for their help and support. And additionally, I would also like to thank the University Undergraduate Mini Grant Committee and University Honors Program for the funding of the project. These are my references, and thank you for listening.
All right, great job. Questions from the audience?